get started here, and I'm going to do some review. I just want to just finish development. Make sure this is recording here. There we go. Okay, development. Okay, the last part of embryonic development is basically the embryonic period. Embryonic development basically refers to the weeks 3 through 8. By week 8, all the organs have been basically laid down. Okay, even though they may not all be functioning, they're all the organogenesis has been complete by week 8 of development. So embryonic period begins at the 3rd week of development and then continues through the 8th week. That's what we call the embryonic period. The blastocyst has been fully implanted. The inner cell mass becomes differentiated into the bilaminar embryonic disc. The bilaminar embryonic disc will become the germ layers. There's 3 germ layers. And the extra embryonic membranes have developed. When we say extra embryonic membranes, we're mainly referring to the chorion and the placenta. And during this 5-week stage, 3 to 8, the conceptus is what we refer to as an embryo. So the embryonic period starts with a process called gastrulation. Gastrulation basically means the cells, the germ layers, are basically folding in and producing basically a rearrangement of the tissues and migration of the tissues. That's what we refer to. We refer to it as the trilaminar embryonic disc, which basically is the 3 germ layers. Okay, the 3 germ layers will become all the major organ systems in the process of organogenesis, and that's by the end of week 8. Okay, and by the way, that is that's a test question. Okay. On the final exam. The placenta also forms that period, and it begins to provide nutrition and oxygen to the embryo and remove waste. Remember, the placenta takes over the function of providing hormones for the remainder of the pregnancy. The remainder of the pregnancy. Remember, the corpus luteum has still survived up until this point. Remember, the corpus luteum persists for about 3 months after fertilization. The corpus luteum will regress and deteriorate after 3 months. Okay, and then the placenta takes over. As the major hormone producer. For the rest of the pregnancy. And so the placenta provides nutrition, oxygen, nutrients, and also removes waste products from the fetus, like the ammonia, the urea. The fetus is still producing waste products, but the placenta will take over that function, mainly through the connection that it has with the mother's blood supply. So let's look at some of the events that occur that lead up to the formation of the 3 germ layers. In week 3, the bilaminar embryonic disc now has 2 cell layers. Those are called the epiblast and the hypoblast. Okay, the epiblast and the hypoblast. These are your 2 major cell layers. The primitive streak forms a thin groove on the dorsal side of the epiblast. It elongates and produces what they call the cephalic caudal or head-tail line of the embryo. It kind of gives it length, and it establishes the head and tail regions, okay, for the future brain and buttock regions, okay. And it also provides a right and left side. This is where your bilateral symmetry evolves. Bilateral symmetry. And then your dorsal and ventral surfaces are going to basically evolve as well. So gastrulation will begin after the primitive streak has completed its development. The first cells then migrate and become the inner germ layer. The endoderm will then become the lining of the GI tract. Respiratory tract, and a GU tract, genital urinary tract. It's mainly a lining surface, okay? Next, the cells then migrate to position between the epiblast and endoderm, and it becomes the middle layer, the mesoderm. The mesoderm becomes all the tissues, becomes all of the connective tissues of the body. You know, muscles, bones, skeletal, blood. And the remaining cells of the epiblast form the outer layer, the ectoderm. The ectoderm becomes the integument, the skin, and the nervous system. Most of the nervous system will form from the ectoderm. Okay. The neural plate and the brain and the spinal cord. So by the end of week 3, all 3 primary germ layers have been formed. Okay, and this is where all the tissues will derive. The embryo has now become a bilaminar embryonic disc. The ectoderm and the endoderm are tightly packed, and they surround a loosely organized mesoderm in the middle. So here's your gastrulation and your formation of your germ layers and so basically, if you look at this diagram, here's the amnion, here's the amniotic cavity on the inside. Okay, here's the yolk sac. The yolk sac, remember, becomes the first blood cells and the gametes. And of course, the chorion basically is on the outside here. Okay, so here's a all right, so let me turn my light on. Okay, so here you've got your cut edge of your amnion. This is the caudal end. This is the cephalic end. So early in week 3, the primitive streak forms on the surface of the epiblast. So you've got the epiblast cells migrating into and then under the primitive streak. So this is a cross-section of the embryo showing the formation of the endoderm by the end of week 3. This is the epiblast cells up here. Here's the primitive streak, and this is the hypoblast. Okay, so this would be the developing endoderm, and this is the ingression that's occurring by the arrows pointing inwards, where the cells basically fold to the inside. So this is another cross section showing you the epiblast cells on the right, migrating between the endoderm and the epiblast, and which then becomes the mesoderm. This is the mesoderm in the middle here. And by the end of week 5, excuse me, it's a little bit larger. Okay. Okay. By the end of week 3, all 3 layers have formed. So that's what we call a trilaminar embryonic disc. Okay. So basically the flattened embryonic disc will change shape during the 4th week. 2 types of folding occur, and it makes the body cylindrical. That's where we have the cephalocaudal folding, where it occurs in the head and tail regions, creating the future head and the buttock regions of the embryo. And then you get your lateral or transverse folding. And that's where your left and right sides of the embryo curve and fold toward the midline. And that's what produces your bilateral symmetry, a right and left side. So anyway, all these sides move close together. As these sides move closer together, the yolk sac is then pinched off, and then it leaves just a small area of endoderm called the vitelline duct. And also the transverse folding during this time becomes the future trunk region, and almost immediately forms the primitive gut, which later becomes the digestive tract inside the abdominal cavity. You know, we're basically, as humans, we are what we call true coelomates. True coelomates, in that we have a true body cavity. True coelomates. You know, we have a cavity within a cavity. You know, the GI tract, the GI cavity is within the peritoneal cavity. Okay. So this is basically the gastrulation and finishing of the germ layer development between weeks 3 and 4 of development. Okay. So here's your cephalic and caudal regions folding in on themselves. So the 3rd week, you got your 3 germ layers, your ectoderm, mesoderm, and your endoderm. Ectoderm on the outer region, mesoderm in the middle region, and then the endoderm becomes the linings of your GI tract and respiratory tract and the genital urinary tract. Here's the future umbilical cord, the allantois. The allantois will make the blood vessels, the umbilicus, and the yolk sac, of course, like we said, produces your first blood cells and your gametes, which will then later migrate to the ovaries and testes. Week 4, you see the folding occurring, and it basically goes from this shape to this shape. This is the yolk sac. This is the red is the mesoderm, and then the blue is going to be the outer amniotic cavity. And then by late in week 4, you've now got a primitive gut, a vitelline duct, which is a remainder of the umbilicus, and then the yolk sac. As you can see, the yolk sac will be pinched off later. Okay, early heart formation, where you see that little red area. And now, when the left and right sides fold in on the midline, then you get, you can see the mesoderm in the middle, ectoderm on the outside, and then eventually by week 4, the folding continues, and then by late in week 4, you can see the endoderm being laid down. Okay, and the amnion, of course, is out here, the membrane, amniotic cavities in here in blue, and then here's the start of the neural tube, and the red would be the mesoderm. Okay, so this is the embryonic folding and the developmental changes that occur between weeks 3 and 4 of development after fertilization. So the 3 primary germ layers will then differentiate into the organs and the organ systems. That's what we refer to as organogenesis. Okay, the 3 germ layers will become all the tissues in the human body. Okay, when organogenesis begins, the primary germ layers have already undergone cephalocaudal, head, tail, and transverse folding. The transverse folding produces the bilateral symmetry, and they're beginning to differentiate. Remember, differentiate means to specialize, produce a given function. Embryo is still very small. It's only a half an inch, 0.6 inches, 15 millimeters. When the embryonic period ends at the end of week 8, the embryo will be approximately 25 millimeters or 1 inch in length, and will have recognizable organ systems, even though they may not all be functional. Some of the organ systems will begin to work. So there's a test question that asks, by the end of week 8, all of the organs have been laid down. Okay, by the end of week 8, all of the organs have been produced. That's a test question. So the ectoderm will become the epidermis of the skin, and it also is responsible for forming the majority of the nervous system and the sense organs. So, and that comes out of the neural plate. The first major event of organogenesis is called neuro. Okay, this is a test question. See, what is the first event of organogenesis? And that's called neurulation. Okay, the ectoderm will thicken and form the neural plate, and it will fold inward and deepen. The edges then fuse to form the neural tube, and then the neural tube then pinches off from the ectodermal layer. And then the anterior of the end of the neural tube becomes the brain, and the remainder of the neural tube becomes the spinal cord. So this will be the basis for the central nervous system and center. And that makes sense that would be the first organ system to develop, because the central nervous system commands the body. By the end of the 4th week, the anterior end of the neural tube has developed into the 3 enlarged areas called the 3 primary brain vesicles, the forebrain, midbrain, and hindbrain. Remember that eventually those 3 primary brain vesicles will become the secondary, and then the mature brain regions. The cerebrum, cerebellum, brainstem, and so forth. But anyway, the neural crest cells between the ectoderm, between the layers of the ectoderm that form the epidermis and the neural tube, those then will migrate throughout the embryo, and they give rise to the cranial, spinal, and the sympathetic ganglia, and their associated nerves, the nerve plexuses, like the gray rami and the white rami. That's what we mean by the associated nerves. The gray rami, communicantes, all those connections within the autonomic nervous system, and the white rami communicantes. The white rami. Those parts of the organ systems that we learned back in chapter 14. Pigment cells of the skin, cells of the adrenal medulla. Remember, the adrenal medulla is really part of your sympathetic nervous system. It works conjointly with the sympathetic nervous system, as well as your connective tissue cells. So the mesoderm cells underneath the primitive streak form the notochord. The notochord becomes most of the vertebral column, which surrounds the spinal cord. It serves to support and organize the embryo around the central axis. The remnants of the notochord remain in the intervertebral disc because the notochord will be surrounded by the vertebral bone during development. The mesoderm on either side of the notochord will form these block-like structures called the somites, and each will have three regions. The sclerotome becomes the vertebral bones and the ribs. The dermatome develops into the dermis layer of the skin, and the myotome becomes the skeletal muscles. Remember, derma means germ. Derma refers to germ layer. Your skin. Of the ectoderm. And myotome, myo refers to muscles, so this becomes just most of your skeletal muscles. And then on the lateral sides of the somat, on the lateral sides of the somites are clusters of mesoderm called the intermediate mesoderm, and the sheets of the lateral plate mesoderm. The intermediate mesoderm form the gonads and the kidneys, ovaries, testes, and kidneys. These develop together. Okay, and the lateral plate mesoderm becomes the spleen, adrenal cortex, most of the cardiovascular system, the heart, blood vessels, the serous membranes of the body cavities, the pleural membranes in the lungs, the pericardial membranes in the heart, and the peritoneal membranes in the GI tract, as well as the connective tissue components of your limbs. Okay. And then the endoderm forms the cavity of the middle ear and the auditory tube, the fringe of the tympanic tube. And the endoderm then becomes the internal epithelial layers of the digestive, respiratory, urinary, and reproductive systems. So it's mainly the lining epithelial layers of your ventral body organs, like the lungs, like the respiratory system. The digestive tract. Digestive tract. And the genitourinary system. And the genitourinary system lining. Okay, so there's going to form several glands and accessory digestive organs, including the thyroid gland, the parathyroids, the thymus. Remember, the thymus is for immune system development. The palatine tonsils, and the majority of the liver, the gallbladder, and the pancreas. Remember, the pancreas and liver and gallbladder are accessory organs of digestion. Okay, these are your accessory organs of digestion. And we're still talking about organogenesis. Okay. So by the end of the embryonic period, which is week 8, Organogenesis has completed. In 4 weeks, the embryo has 3 primary brain vesicles and somites. By the 8th week, the embryo looks essentially human, even though it has different body proportions. And during weeks 3 through 8, also the embryo is very sensitive to what we call teratogens. A teratogen is anything that's harmful to the fetus that's man-made. Any agent or any substance, like a drug or medication, like an illicit drug or medication prescribed by a healthcare professional, that is destructive to the embryo. So the embryo is very vulnerable at this time. So that's the reason that expected mothers have to be especially careful in that first trimester of their pregnancy, because that's where the birth defects are going to occur. So here's the mini MRI showing you an embryo at week 4. You can see the buds of the arms and the leg buds. The eyes are basically laterally positioned still. They're on the lateral side of the head. They haven't migrated to the middle yet. Forebrain appeared. Remember, the forebrain will become the cerebrum, the telencephalon, and then you can see where the ears are going to be located. Okay, and these would be the somites. Okay, which will become the cleratome, epitome, and myotome. Okay. Oh, I forgot to show you the week 8. This is a fiber optic view at the end of week 8. You can see the eyes are moving more immediately. There's the nose, forebrain, legs, arms, elbow, fingers. Here's the umbilicus, the umbilical cord, containing the lifeline to the placenta. So there's your 3 germ layers: ectoderm, nervous system, epidermis, hair follicles, erector pili muscles, nails, epithelium of the nasal, oral, and anal canals, lens and cornea of the eye, internal eye muscles, internal and external ear, salivary glands, adrenal medulla, pituitary and pineal glands, and the melanocytes. Mesoderm, everything in the middle, skeleton, cartilage, and all the connective tissues, skeletal, cardiac, and smooth muscle, adrenal cortex, middle ear, dermis, blood, and lymph vessels, bone marrow, lymphatic tissues, kidneys, ureters, gonads, genital ducts, mesothelium of the abdominopelvic and the thoracic cavities, the linings. And then finally, the endoderm becomes the epithelium of the lining of the digestive tract. Simple columnar epithelium. Simple columnar epithelium. Okay. And also your so anyway, it becomes the lining for your digestive tract. Only exceptions for the endoderm would be the oral, nasal, and anal canals, because that's where you'll find the stratified squamous epithelium. And the respiratory tract. Also epithelium of the urinary bladder and the urethra, as well as the epithelium of the accessory, reproductive, and the digestive glands, all except for the salivary glands. The thyroid, the parathyroid, and the thymus glands. Okay, so those are your 3 germ layers by the end of week 8. Now, the last period of the pregnancy is what we refer to as the fetal period. We've talked about the pre-embryonic period between fertilization and 3 weeks. Okay, that's what we call the pre-embryonic period. The embryonic period, let me just review again the three phases of pregnancy. Pre-embryonic, period. Remember, that's between fertilization and 3 weeks post-fertilization. Okay. Second phase, that's the embryonic period. This is when in the embryonic period, this is still the first trimester, but this is the period that's very vulnerable to carcinogens. That's between 3 and 8 weeks. Post-fertilization. Now we've got the fetal period. The third phase of gestation is what we refer to as the fetal period. This is where you're going to find the most rapid growth. Fetal period is between it basically starts at 9 weeks until 38 weeks. And the termination or the end of the pregnancy and birth. Okay. So the fetal period is week 9 until birth, about 38 weeks. If it's 38 weeks, that means it's full term. You need for the baby to go full term to have all of the organs completely develop, okay, and less complications. So the fetal period of development will extend from the beginning of the week 9 until birth, usually near the end of the 38th week. During the pre-embryonic period, remember the developing embryo receives nutrients from uterine milk secreted from the endometrial glands, as well as digested endometrial cells from the uterine wall. And then the embryo will then continue to receive nutrition from the digestive endometrial cells until the placenta is fully formed by about week 12. And that will be during the fetal period. And then placentation, basically you're forming the placenta, the dish-shaped placenta, which attaches to the uterine wall and to the embryo or the fetus through the umbilical cord. The placenta, remember, is a temporary organ that is shed after the infant is born. It's what we refer to as afterbirth, but it's where the site of exchange of oxygen, nutrients, and waste products between the mother and the fetus. The placenta also produces the hormones to support pregnancy. Placentation begins during implantation, but the majority of the placental growth will occur in the fetal period. So here's your future placenta over here, okay? Even though these little, this is going to be the maternal blood supply on this side, and then the fetal blood supply is going to be on the right here. Here's the yolk sac, the developing embryo, the amniotic cavity with the fluid inside, and the amnion. And then the chorion is out here. Okay, so the chorion is what we call an extra embryonic membrane because it's outside the amnion. This area is called the deciduous capsularis. Here's the allantois, this area right here, and then this is going to be the placenta, okay, and the yolk sac. So the umbilical cord will connect the center of the placenta to the fetal umbilicus or the belly button. There's two umbilical arteries which carry deoxygenated fetal blood away from the fetal heart to the placenta. One umbilical vein which carries oxygen and nutrients toward the fetal heart. Remember, we usually use the red colors to denote the umbilical vein. So here's the umbilical vein, basically taking oxygenated blood from the placenta to the fetus. Fetus is, of course, going to be and then you have the two umbilical arteries, which are going to carry deoxygenated blood, but they're going to be kind of wrapped around the single umbilical vein. So you'll have your pairs of umbilical arteries basically wrapping around the single umbilical vein. So they'll go in the opposite direction. Okay. So this whole structure is your umbilicus or your umbilical cord. Okay. So the umbilical arteries are going to carry blood in this direction, and then the umbilical vein will carry blood in this direction, back toward the fetus, oxygenated blood from the mother to the fetus. So anyway, the umbilical cord has a jelly-like consistency called Wharton's jelly, which is connective tissue, and it insulates and protects the umbilical arteries and vein. And the external surface is then covered by the amniotic membrane. So when the embryonic period begins, the blastocyst has now been firmly implanted in the uterus, and the chorionic villi have now penetrated the uterine blood vessels to form cavities called lacunae, filled with maternal blood. And they then merge into a single blood-filled cavity called the placental sinus. So once the placenta has been established, then the stratum functionalis of the uterus is called the decidua. Remember, the stratum functionalis is a temporary layer of the endometrium that's shed every month. So the endometrial region that lies beneath the fetus becomes the decidua basalis, and the region that surrounds the uterine cavity forms the decidua capsularis. You know, that picture that I just showed you a while ago. That's the decidua capsularis, but it's because it encapsulates the entire amnion. So this is going to be your chorionic villi. Okay, and here you can see the single umbilical vein, which carries blood back to the fetus, and then the two umbilical arteries, as you can see, they wrap around the single umbilical vein. Okay, but they're going to carry blood to the placenta, so they're going to go in this direction. Okay. And then the umbilical vein, it will go in the opposite direction. Umbilical vein will carry blood to the fetus, okay, because it's carrying oxygenated blood. Okay, it's going to carry oxygenated blood. Whereas the umbilical arteries are carrying deoxygenated blood. These are the, like I said, the chorionic villi, where you get your exchange of blood and nutrients and oxygen and waste products between the fetal blood, which is through the two umbilical arteries, and the maternal blood, okay, which is basically going to become the umbilical vein, which is colored red here. Okay, so on the left here you see the maternal blood supply. The red are the maternal arteries, and the blue is maternal vein, which basically drains the blood from the placenta. So anyway, this will be the chorion in the middle here, okay? Decidua basalis over here on the mother's side. And then all of this blue area is the maternal blood in the placental sinus, okay? All this blue area, okay? So everything that you see inside here is in pink here is the placental barrier. Everything that you see here in pink is the placental barrier. And you still have to have a barrier to prevent any kind of immune complexes from forming, so you don't get autoimmunity. So the placenta is a unique organ because it develops from both fetal, the chorionic villi being an example, and maternal, the decidua basalis structures. It's basically going to be part of the mother, part of the fetus. The villi, the chorionic villi, are filled with blood vessels and that are carrying fetal blood and surrounded by maternal blood in the placental sinus. It will sort of serve as, it looks more like the GI tract villi, okay, like in the small intestine, but it serves as a respiratory system, okay, as well as the exchange of waste products between the fetus and the mother. The blood supplies do not mix at all, though, okay? And that's because of the placental barrier, which is composed of a basement membrane and a thin layer of syncytiotrophoblasts cells, which keep the blood supply of the mother and the fetus separated. So since your maternal and fetal blood supplies are very close, some substances are exchanged across the placental barrier between the fetal and maternal blood supplies. Oxygen and nutrients will diffuse from the maternal blood into the fetus to be transported to the tissues of the fetus, whereas waste products from the fetus will diffuse from the fetus to the maternal blood for removal. Of course, they arrive from the two umbilical arteries. So this is your placental barrier in the middle. Okay, so you can see oxygen and nutrients are going from the maternal blood in the placental sinus, eventually into the fetal blood, in the capillary. Okay, whereas the waste products from the fetus are being delivered to the maternal blood in the placental sinus. So you can see this is the placental barrier, which consists of the syncytiotrophoblasts and the cytotrophoblasts. Okay, and then the fetal basal lamina and the maternal basal lamina form the internal and external boundaries. Okay, and all of this kind of beige colored area or light pink, those are the fetal connective tissues. Okay, these are the fetal endothelial cells that surround the fetal blood in the fetal capillaries. So blood flow, therefore, between the mother and the fetus to the placenta basically will have oxygenated maternal blood that flows to the bassalis layer of the uterus and then through the maternal arteries into the placental sinus. The maternal blood will then touch the placental barrier. That will allow diffusion of substances, but not the passage of blood cells or proteins. So red cells, white cells, immune cells, those all stay within the mother's blood, okay? And once that diffusion occurs, though, the maternal blood, which is deoxygenated and carrying waste, will then flow into the maternal veins and back to the mother's cardiovascular system. So the mother is excreting waste products from the fetal circulation. So the mother is really doing double duty. Is really taking care of two organisms. So your deoxygenated fetal blood then will leave the fetus and then enter the two umbilical arteries, which then flows into the placenta. And then it moves into the fetal capillaries in the chorionic villi, those little finger-like extensions that you see in the placental sinus. These are the chorionic villi, which is where your umbilical arteries and veins are going to meet. Okay, so fetal blood will then pick up oxygen and nutrients and deliver waste by diffusion. But again, remember, there's no blood cells that move through that placental barrier, which is the syncytiotrophoblasts and the cytotrophoblasts. That's your barrier. Also, another feature that allows the fetal blood to be oxygenated is the type of hemoglobin that's present in the fetus. The hemoglobin in the fetus is actually different from the adult hemoglobin. Fetal hemoglobin has a much, much higher oxygen affinity, attraction for the oxygen, than adult hemoglobin does. Okay, so fetal hemoglobin has a greater affinity for oxygen than the maternal hemoglobin, so that will serve to pull the oxygen away from the mother's adult hemoglobin at the placental barrier. Okay, so the oxygenated blood then will leave the placenta and then flow back to the fetus through the single umbilical vein, which is why it's colored red. Remember, the umbilical arteries wrap around the umbilical vein, which they're carrying blood in the opposite direction than the fetal vein, than the umbilical vein. So in addition to nutrient functions, the placenta also is an endocrine organ because it assumes the production of ACG production as it grows. It also results in reduction of the centrotrophoblasts as a result. The corpus luteum, remember, persisted for 3 months after fertilization if pregnancy occurred. Okay, and it mainly persists to provide the hormones for the developing embryo. Okay, so it relinquishes production of progesterone and estrogens to the placenta by the end of the 3rd month of gestation. So the placenta will take over the role as the hormone producer. Also, the placenta produces HPL and placental prolactin, human placental lactogen, and this helps prepare the mammary glands for production of milk, for lactation. Milk production, of course, is what we refer to as lactation. Under the influence of the hormones like prolactin. Another hormone that's produced by the placenta is called relaxin. Relaxin relaxes the body muscles, joints, and ligaments, and this is going to be important in labor and delivery. In delivery of the newborn, it allows for the facilitate stretching during the delivery of the newborn, particularly in the pelvic structures in the mother. And there's a condition called placenta previa, and it's a very dangerous condition that sometimes can occur with birth. It's called placenta previa. Okay, that means part or all of the placenta will actually attach to the inferior portion of the uterus. It doesn't actually separate, and that's very near or even covering the cervix. Most cases will result in a live birth, but they may cause spontaneous abortion or a premature birth if it's severe. It's very dangerous to the mother because as the uterus stretches to accommodate the fetal and placental growth, the placenta may actually start hemorrhaging or become infected, and that increases the risk of maternal mortality. In the early part of the 20th century, a lot of the cases of death of the mother through birth are because of placenta previa. Typically, cesarean section, surgical removal of the fetus, through the abdominal wall, is the preferred method of delivery in cases of placenta previa. And of course, the OB doctor, the healthcare professionals will be able to recognize this early on in the ultrasound, in the sonogram. So fetal development, this is where you get your rapid maturation of your tissues and organs. Fetal growth is extremely rapid, and fetal size will dramatically increase from week to week. Okay, so what they use when they're using the sonogram, they're basically taking fetal measurements of length, and those include what they call the crown rump length, okay, or the crown heel length. The crown rump length, of course, is the distance between the head and the buttocks, okay, or the head and the heels of the feet. So the embryo, about 25 millimeters, 1 inch in length, can grow to be as large as 50 centimeters, 19.7 inches, crown heel length during the fetal period. Weight increases also most obviously in the last 8 weeks of development. Average weight of a full-term fetus usually ranges from 3.2 to 3.5 kilograms, or between 7 to 8 pounds. Remember, for every kilogram, there's 2.2 pounds. So 3.2 times 2.2 would be about 7 to 8 pounds. Month 3, the body lengthens as the head growth starts to slow down. The upper limbs will grow to their birth length, and that's when you get your ossification beginning in most bones. From ossification is the mineralization that produce the hydroxyapatite crystals. Eyes are also well developed, but they're located on the lateral side of the head. The eyelids are fused, the bridge of the nose will develop, and the external ears are present, but they're not prominent yet. You can just see the earbuds. The genitals are distinguishable as male or female by month 3. By the end of the 3rd month, the CRL is about 3.5 inches, 9 centimeters. Month 4, you get even more rapid body growth. The lower limbs will lengthen, joints are forming, the skeleton continues to ossify. The fetus now has nipples and some hair by month 4. At that point, the kidneys are well formed, the digestive glands are forming, and the heartbeat can be heard with the stethoscope by the 4th month. The fetus also begins to develop reflexes and will startle and turn away from loud noises and bright lights. It also experiences pain. The fetus also experiences pain. Approximate CRL is about 14 centimeters by the end of the 4th month. At month 5, the growth will slow and the lower limbs will achieve their correct proportions relative to the body. You start seeing hair growth on the head. The skin is now covered by downy hair called lanugo, which will eventually disappear. And you'll see these little secretions, these little white to gray secretions, which are basically composed of shed epithelial cells and sebum called Vernix caseosa. And this is important because of the amniotic fluid. The lanugo and the Vernix caseosa basically cover the skin to protect the skin from the amniotic fluid. At month 5, there's formation of brown fat, and that helps with production of heat after the fetus is born. The mother can also feel movements because of a phenomenon called quickening, because the skeletal muscles are beginning to contract in the fetus. The approximate CRL is about 19 centimeters, which is about 7.5 inches by the end of the 5th month of the fetus. So this is basically between 6 and 15 weeks of fetal development. So this is a fiber optic view on the left of a fetus at the 3rd month. Okay. You can even see part of the genitals, the fingers, the ears, the forebrain, which is going to be the cerebrum, the feet, the knees. And then on month 5, this is a fiber optic view. You can actually even see the fingernails. Okay. The nose, the nostrils, the lips. Okay. The eyes are more frontally located. So this is at month 5. Okay. Looks fairly human now. Okay. These are the actual sizes, okay, of the fetus. This is from a fiber optic camera that was placed in the embryo from a live birth. The fetus at month 6 will still really start gaining significant weight. The eyebrows and the eyelashes appear. The eyelids have been fused since the 3rd month, are now partially open. The skin is wrinkled and translucent, and the lungs start to produce surfactant. Remember, surfactant is key to survival if the infant is born prematurely. And if it is born at this 6th month, it won't have much surfactant, okay? The CRL is about 9.1 inches, 23 centimeters by the end of the 6th month of fetal development. Month 7, this is when the eyelids start to open completely. Fat is now deposited in the subcutaneous tissue, so the skin becomes slightly smoother, but still it's fairly wrinkled and red. The fetus usually turns upside down during the 7th month, assuming the vertex position. That's why they always have to get those sonograms at the 7th month and when you're getting prenatal care. In males, the testes begins to descend through the inguinal canal and into the scrotum. And by the end of the 7th month, the CRL is about 11 inches, almost a foot, 28 centimeters. The 8th and 9th months, okay, nearing the end, remember 288 days, fetal neurons then form networks, blood cells form in the bone marrow, the digestive and the respiratory systems complete the development during the 9th month. The fetal skin now is less wrinkled, and the nugo is basically shed. In males, the testes will complete their descent into the scrotum. This is the time period when if the testes do not descend on both sides, remember that's called cryptorchidism. Okay, and that makes the male infertile. The approximate CRL is 30 centimeters, almost 12 inches by the end of the 8th month, and 36 centimeters, 14 inches by the end of the 9th month. So in about 266 days after fertilization, the fetus is now considered full term and is ready to be born. The scalp will generally have hair. The fingers and toes have well-developed nails. If the fetus is born before that time, the tissues and the organs may not have developed completely. So that's the reason you have to wait to full term for birth. The fetal circulation and the cardiovascular system will change rapidly soon after birth, mainly to adjust to life without a placenta and into a different environment. Most of the conspicuous changes in postnatal state occur mainly in the cardiovascular system. Some of these changes we've seen when we looked at the cardiovascular system. We'll just kind of review that. Remember the unique cardiovascular structures that are present during prenatal development include the 2 umbilical arteries and the umbilical vein, and your 3 circulatory shortcuts called the vascular shunts, which are the ductus arteriosus. The ductus arteriosus, remember, is the connection between the pulmonary trunk and the aorta, okay, which allows blood to move from the pulmonary trunk directly into the aorta, so it bypasses the lungs. The other vascular shunts are the ductus venosus, which allows blood from the umbilical vein to bypass the liver, and it's connected then directly to the inferior vena cava, and then flows into the right atrium of the heart. Remember, the liver is not necessary in the fetal period of life because, of course, the nutrients are derived from the maternal blood supply while the digestive system is forming. And then the third vascular shunt is called the foramen ovale, which is the hole in the interatrial septum that directly connects the right and left atria. It allows the blood to skip ahead and avoid the pulmonary circulation by moving directly from the right side of the heart to the left side of the heart and then into the systemic circulation of the fetus. So those are your 3 vascular shunts: ductus venosus, foramen ovale, and ductus arteriosus. So after birth, the shunts and the umbilical vessels will close, and the normal circulatory system begins within a year in most infants. Flaps in the interatrial septum will seal, and the foramen ovale becomes a depression called the fossa ovalis. And then you have more resistance and pressure changes in the pulmonary trunk and aorta. Those will cause the ductus. Remember, birth occurs with the lungs expanding and pressure changes. And that will cause a ductus arteriosus, the little connection between the pulmonary trunk and the aorta, to close. And that usually is complete by 3 months after birth. The remnant of the ductus arteriosus is the permanent ligamentum arteriosus. That's where you see that ligament between the pulmonary trunk and the aorta. And then the ductus venosus will degenerate and become the fibrous ligamentum venosum. And then the umbilical arteries become the medial umbilical ligaments, and the umbilical vein becomes the ligamentum teres, also called the round ligament of the liver. It's on the ventral side of the liver where you see the 4 lobes. So these are your unique cardiovascular features, like the ductus arteriosus, which becomes the ligamentum arteriosum. Remember, that's the structure that you see right here between the pulmonary trunk and the aorta. It just allows blood to basically bypass the pulmonary circuit, okay? Because remember, the lungs are collapsed. And then the foramen ovale, the connection between the right and left atrium, becomes a depression called the fossa ovalis, located in the wall of the right atrium. And then the ductus venosus, which is the continuation of the umbilical vein that bypasses the liver to directly enter the inferior vena cava, the ductus venosus becomes a ligamentum venosum. This is the ligamentum venosus. And then the umbilical vein becomes the ligamentum teres in the adult. And then the umbilical arteries become the medial umbilical ligaments in the adult after birth. So prematurity, when an infant is considered premature if it's born more than 3 weeks before full term being 38 weeks. More than 12% of babies born in the United States each year are premature. The earlier the birth, the more complications the infant is likely to experience. Most commonly, premature infants suffer from difficulties with respiration, digestion, and thermoregulation because those organ systems have not completely developed. So your most common respiratory problem is what we call infant respiratory distress syndrome, or IRDS. It develops in infants born before 24 weeks because the surfactant is not produced in the alveoli of the lungs until this time. Without surfactant, the alveoli will collapse during exhalation, and they're difficult to reinflate due to the alveolar surface tension. So synthetic surfactants can help premature infants breathe until their lungs can develop sufficiently. Also, premature infants often have minimal sucking and swallowing reflexes, and so they require feeding tubes. Also, they have an underdeveloped liver, which cannot produce enough clotting factors or albumin. So they bleed very easily and may develop swelling, edema, because of the lack of albumin. Hypothalamus also is not completely developed, so they cannot really maintain a constant body temperature. And so they have to be kept warmer, usually in an incubator. Many years ago, they actually used ovens in homes to serve as an incubator when they didn't have hospitals that allowed this. So premature birth is probably the most common cause of infant death in the United States. We have a lot of advances in neonatal care. About 30% of infants are born at 23 weeks, 50 to 6% in 24, 75% are born at 25 weeks, and more than 90% are born in 27 and 28 weeks. So infants who do survive, which most of them do, because of the advances in neonatal care, but they generally will often face lifelong issues like cerebral palsy, learning disabilities, as well as hearing and vision losses. Okay. So that's basically the end of the lectures. I want to go through some. Oh yeah, they used to use ovens. I have a friend that was a friend of mine many years ago. I guess he's in his probably in his 80s now. But when he was born in the 1930s, okay, and they actually used ovens to basically keep them warm, okay, and they kept them alive though. But he's still alive today. He does have, because he was born so prematurely, he has problems with hearing and balance, and a lot of times that happens. But he's still alive. He's in his 80s. So, even though we didn't have sophisticated care back then, people, especially, they knew how to keep the baby alive. Okay. So anyway, we're going to start looking at some. Let me go through some Jeopardy questions. We'll do chapter 26 first, then we'll do chapter 25. Which tube carries sperm from the scrotum to the abdominal pelvic cavity? Urethra, ductus deferens. Ductus deferens is the same thing as the vas deferens. Well, hopefully you said vas deferens. Vas deferens is correct. Which structure is not associated with the testes? Remember, the tunica albuginea is associated. Tunica vaginalis. The tunica vaginalis is the same as a continuation of the peritoneal membrane. Veniform venous plexus, remember that helps to cool the testes. Okay, so yeah, D is correct. D is correct. This gland is located just below the urinary bladder.
The prostate gland.
Exactly, prostate gland. What part of the penis contains the urethra? Remember the corpora cavernosum are paired, the spongy, exactly. The B is correct. Remember the corpus spongiosum is singular, the corpora cavernosum are paired. The cavernosum are dorsal, the corpus spongiosum is ventral. It basically completely surrounds the urethra. The prepuce is the covering, the crura are the body folds. Which of the following is not an accessory gland that contributes fluid to the semen? Well, one, make sure you know what they are. Remember, there's three glands: seminal vesicles, prostate, and bulbourethral. Remember, the seminal glands, I call them the seminal vesicles. Remember, they provide about 60% of the secretions. Prostate about 30%. And then bulbourethral gland about 5%. Okay. Remember, the bulbourethral gland mainly serves to neutralize any acidic urine that might be present. Prostate gland provides the coagulating proteins and the enzymes, and the seminal gland provides most of the nutrients like fructose and ATP. The epididymis is not an accessory gland. Remember, the epididymis is only a storage organ for the sperm. That's where a lot of sperm mature. Okay, the epididymis is a storage organ for the sperm. The luteal portion of the ovarian cycle lasts how many days? The luteal portion would last about 14 days. The entire menstrual period would be 28 days. The follicular phase and the follicular phase and the luteal phase would be about 28 days. But the luteal phase alone is exactly 14 days. Okay, so it lasts exactly 14 days, and that's an exact number. Okay, you can start counting the days, and it's exactly 14. Okay, whereas the follicular phase, the other half of the ovarian cycle, the follicular phase is somewhat variable. Follicular phase may be between maybe 13 to 15 days, but it's on the average it's 14. But it may vary between 13 and 15. But the luteal phase is always 14 days. because it occurs right after ovulation because of the LH surge. Which of the following produces progesterone? Hopefully you're thinking corpus luteum. Corpus luteum mainly produces progesterone, okay? It does produce a little bit of estrogen, but it's mainly progesterone. Remember, progesterone is the hormone of pregnancy, and it's also the hormone that's secreted in the second half of the ovarian cycle. Progesterone, okay. What events may occur in the fallopian tube? Yeah, you sure don't want that to happen, but it sure does occur, unfortunately. And of course, fertilization usually occurs in the oviduct. Endometrial sloughing, that occurs in the vaginal tract. Okay, so answer would be D, both A and B. What are the functions of luteinizing hormone? Usually, if you don't know exactly what the answer is, you can kind of rule out what's wrong, okay? LH comes from the anterior pituitary, okay? So it can't be coming from the corpus luteum if it comes from the anterior pituitary. So you can already mark that one out, okay? HCG is the hormone of pregnancy. This is what we test for because this is produced by the placenta, by the newly developed conceptus. Okay, so HCG is the basis for the urine pregnancy test. Okay, and of course LH is formed in the brain out of the placenta. LH basically functions to initiate ovulation. Okay, it basically is what we call the LH surge. Right in the middle, remember LH is very low at the beginning of the 28-day cycle of the follicular phase, but right at the very middle, it surges like this. It goes way up like this, right about day 13, okay, of the menstrual cycle, day 13. Okay, when you get that's called the LH surge. And at the same time, the FSH is also pretty high, okay, but it's this LH surge along with the estrogen increase that basically stimulates ovulation, okay, release of the egg. And really what happens when you take birth control pills, oral contraceptives, are basically combinations of estrogen and progesterone, okay? And what they do is by negative feedback, they inhibit this release of LH. And when you don't have this LH surge, you don't get ovulation, okay? But it initiates ovulation. Basically, during labor, the main hormone that's working is oxytocin, okay? Oxytocin is at work here, but that's what we call a positive feedback. The positive feedback loop of oxytocin hormone contracts the uterus during labor, and it makes the labor contractions faster and stronger, okay? The time between contractions will progressively get shorter, and the force of contraction progressively gets stronger through a positive feedback. The more, the stronger the force of contraction, the more oxytocin is produced. Okay, that's called a positive feedback mechanism. Most oral contraceptive birth control pills contain what? If you take OCPs, oral contraceptives, what's in those pills that produces a negative feedback? Anybody want to give me an answer on this one? Yeah, it's actually estrogen. It's actually estrogen. It's actually a combination of estrogen and progesterone. Okay, and remember, the way birth control pills work is estrogen and some progesterone negative feedback inhibit the LH surge, and they also inhibit FSH as well. And with no LH, no ovulation. And that's what we call negative feedback. And that's how birth control pills work, okay? That's the reason they're over 99% effective. Over 99% effective. If you're on birth control pills, you won't get pregnant most of the time. You have that 1%, but usually they find out that the patient has not been taking them reliably. They have to be taken every day, okay? But they work by negative feedback by inhibiting the release of LH, the LH surge. The menstrual phase of the uterine cycle typically lasts how many days? Remember the 3 phases of the uterine cycle, just kind of review here. 3 phases are menstruation, we'll call menses. And you've got your proliferative phase. Remember, we're talking about the uterine cycle, okay, or the menstrual cycle. Proliferative phase will coincide with the follicular phase of the ovarian cycle. And then the 3rd phase is the secretory phase. Okay, the secretory phase of the uterine cycle. Uterine cycle is also called the menstrual cycle. Okay, coincides with the luteal phase of the ovarian cycle. Okay, so those are your 3 phases. Menstruation lasts for about 5 days. Okay, proliferative phase basically is 6 through 14. 6 through 14 days. And then the secretory phase will last exactly 14 days. Exactly. Okay, the secretory phase will last 14 days. Okay. Okay, but usually the proliferative phase, since you got 5 days of menstruation, which is days 0 through 5, the proliferative phase will start on day 6, so it's going to basically be about 9 days. Proliferative phase will be about 9 days. Most proliferative phase is usually 9 to 10 days. 6 through 14 would be about 9 days. Okay, and then but the secretory phase, it's usually exactly 14 days. So that basically makes your 28-day cycle. 14 plus 9 is 23, and then 5 is 28. Okay. Proliferative phase about 9 days because it's right after menstruation. Why does menstruation occur? Menstruation occurs because menstruation occurs because of the falling levels of progesterone from the corpus luteum. and estrogen only, okay, in the last half of the menstrual cycle, when the corpus luteum is degenerating. If fertilization does not occur now, then the corpus luteum will start to degenerate and quit making progesterone and estrogen. It mainly makes progesterone, but some estrogen. And when you don't have progesterone and estrogen, then you don't have a uterine wall. The endometrium won't be able to persist, so the endometrium will start sloughing off. So that's why we have menses. Menstruation occurs because of the falling levels of hormone levels, estrogen and progesterone, mainly progesterone, because progesterone is what makes the endometrial lining secretory. So 5 days. What male structure is constricted during enlargement of the prostate? What goes through the prostate gland? Well, the continuation of the vas deferens is called the ejaculatory duct. So the ejaculatory duct is constricted. The reedy testes is behind the testes. The seminal vesicles, they don't even lie in the testes, they lie on the bladder. Okay, and the vas deferens becomes the ejaculatory duct when it goes through the prostate. So the ejaculatory duct, and the other structure they don't mention is the prostatic urethra. So the prostatic urethra also is constricted. They didn't mention that one. It's not given as a choice. That's what causes hesitancy when men get an enlarged prostate. But the ejaculatory duct, where does sperm production occur in the testes? Well, again, rule out the questions the answers that don't make any sense. Yeah, the seminiferous tubule, that's correct. That's correct. Remember, the reedy testes is really behind the testes. Interstitial cells, remember, these are what produce the hormone testosterone. Epididymis, as we said, lies on top of the testes. And basically, it stores sperm for about 2 to 3 months after they're produced. Implantation occurs within what layer of the uterus? Right, exactly. That's right, endometrium. What forms immediately after ovulation in the ovarian cycle? The Graafian follicle is the mature secondary follicle that releases the egg. The Graafian follicle is what releases the egg. So that's really not a choice. Primary follicle, remember, primary follicles are in the outer cortex, and these become secondary follicles. Primary follicles contain a primary oocyte, okay, but they're early in the development. So there's only 2 possibilities: corpus luteum or albicans. So which of those is the answer? Corpus luteum. Corpus luteum is the answer to that one. Remember, the corpus albicans is a corpus luteum that is degenerated. Corpus luteum that has degenerated and is just scar tissue. So there's no hormones produced by the corpus albicans. That's the reason menses occurs in the first 5 days of menstruation, because the corpus albicans is not producing any hormones. The corpus luteum is degenerating into a corpus albicans, no hormone, that causes the menstrual fluid to be discharged. Okay, but the answer would be the corpus luteum. It follows immediately after ovulation. And the corpus luteum comes from the Brachyan follicle that released the egg. It releases the secondary oocyte. And so that's the only time you have a follicle becoming an organ. Estrogen and progesterone are at their lowest concentration during which phase of the female reproductive cycle? Secretory, ovulatory, luteal, or menstrual? Well, that's really the reason we have menstruation is because of the low levels of estrogen and progesterone. Okay. So it would be the secretory would be the highest secretion of progesterone. The secretory phase would be the highest levels. Ovulatory, during ovulation, they're also high here also. You know, you get a lot of estrogen. High estrogen is present, along with LH and FSH, okay? Luteal phase is going to be the progesterone levels are high. Progesterone levels are high. That's because the corpus luteum is making progesterone. So, but at the menstrual phase, you hardly have any hormones, and that's why menstruation occurs. This is why the endometrium sloughs off during menses, okay? There is no hormone like progesterone to support the endometrium. The reason you have an endometrium that becomes secretory is because of progesterone. And when you don't have progesterone, we don't have an endometrium. And that's what causes menses. So the menstrual phase, primary spermatocytes are produced during which process? Meiosis 1, meiosis 2, mitosis. Again, you can rule out the wrong ones. Okay, right off the bat, you know mitosis doesn't even occur with reproduction. Okay, well, it does occur. It occurs in the production of the primary spermatocytes, but it doesn't have anything to do with well, hold on just a second. With primary spermatocytes, okay, primary spermatocytes are produced from spermatogonium. So they would be, primary spermatocytes are produced during mitosis. Okay, so that would be the answer. So the way it happens is spermatogonium or diploid, they become primary spermatocytes. And that involves mitosis. Okay? This involves mitosis. Okay? You're just duplicating the primaries for the DNA. Okay, and then the primary spermatocytes which are diploid, they're 2N, they have 46 chromosomes. They become secondary spermatocytes. So that's what we call the reduction division. Those would be haploid. They'd have the 1N chromosome number. And that's through meiosis 1. Okay. Meiosis 1. Okay. And then the secondary spermatocytes which are haploid, 1N, or we'll call the haploid number. they become spermatids through meiosis II. And for meiosis II is just separation of the sister chromatids, though. Okay, separation of sister chromatids. That's what occurs in meiosis II. That's the reason meiosis II is similar to mitosis in the events. So the only time mitosis occurs is during the development of the primary spermatocytes from the spermatogonium. Okay, so that's what we refer to as mitosis. Mitosis will allow spermatogonium to become a primary spermatocyte. And then meiosis I occurs between the primary and the secondary. And then meiosis II occurs between the secondary and the spermatid. Okay. So the answer would be C. Where in the body are sperms stored? Correct. That's correct. Where do sperm acquire their motility? Mainly the epididymis also. That's where they mainly develop the ability to be motile. Okay, the flagellum is fully developed. Remember, spermatogenesis requires which of the following hormones? Remember, you've got to have LH, FSH, and testosterone. All three of those hormones are needed. Okay, you need all three of those. If without LH, you won't have the production of testosterone. Without FSH, you don't have the androgen binding protein that keeps the testosterone in the area for proper development of spermatogenesis. Okay, and you need testosterone even for spermatogenesis to even occur. So you need all three hormones. Erection in the male is controlled by the parasympathetic nervous system, while ejaculation is controlled by the sympathetic. Parasympathetic is erection, sympathetic is ejaculation. Ask me why, I don't really know. Wish I did. I've always tried to find that out, why sympathetic is responsible for ejaculation and parasympathetic is erection. Don't know though. Don't really know, and I haven't found out from any doctor or anybody that's researched this. Nobody's really given a satisfactory explanation to that. The superior part of the uterus is called remember there's three parts of the uterus. You got the fundus. Yeah, fundus, that's right. Fundus is right. Good. What structure has fimbriae? Uterine tube are the same thing as oviducts. Yeah, the fimbriae would be a part of the uterine tube. The fimbriae, the uterine tube looks like this. Here's the uterine tube coming down like this. And it's got these little finger-like projections. And these are ciliated. And then what they do, they produce these little canals. Okay? And then here's the ovary underneath. And then here's the follicle, grafting follicle, fix and release an egg here. Okay? So there's little cilia at the very end and inside the canal that sweep back and forth. And what they do is that secondary oocyte is released really into the peritoneal cavity, but they're swept up immediately by these fimbriae. Okay, they go straight up into the fimbriae. because of the current that's produced here. It's kind of like a current in a bathtub that picks up something, okay, as you open up the drain plug, okay. But it's basically cilia that beat back and forth that line the fimbria, and then they sweep it up into the oviduct. But they're part of they're in the uterine tube, okay, the oviduct. Follicle cells are part of the tunica albuginea, corpus luteum, primordial follicle, or vesicular follicles. Well, the follicle cells are actually part of the primordial follicles as they develop. In the nursing mother, milk is stored where? These are also called the ampulla, like Tiffer's ampulla. Yeah. But actually, it's D. It's the sinus is where the milk is stored, and then it's released through the duct, and then they're produced by the glands. Remember, the glands are compound alveolar. But anyway, I just want to mention these are also referred to as ampulla. They're just, it's basically where the milk is stored. And that's usually when a mother is, after she's delivered, they're real sore because of the sinuses storing all that milk that occurred in the last trimester. The uterus is anchored to the anterior body wall by the that's right. Round ligament. That's right. The broad ligament is basically, here's your oviduct. And here's the fimbriae down here. Okay, then here's the ovary. Okay, so the broad ligament is all this ligament that you see down here. So it's all the way up to the oviduct. But all of this is the broad ligament. Okay. Suspension ligament basically suspends the ovaries. Anyway, that was the round ligament, anterior body wall. Ovulation is stimulated by the basically the ovulation, LH. Fertilization typically occurs in the what part of the uterine tube? Usually the ampulla of the uterine tube. But it's always in the, hopefully it's in the uterine tube. If it's not in the uterine tube, you may have problems, okay? Hopefully it's not in the vagina or of course it can't be in the vagina. It can't really be in the ovary either. And it really can't be in the uterus because it wouldn't be able to make it the sperm wouldn't be able to make it that far. So it only basically occurs in the uterine tube. Okay, so that's the answer. But it's the ampulla portion, the widening of the uterine tube. Immediately after ovulation, estrogen and progesterone are secreted by the corpus luteum. During part of the normal menstrual cycle, when the levels of progesterone decrease, what will occur? Usually when you don't have estrogen and progesterone, you get menstruation, okay? Ovulation is when the eggs release. Fertilization is when the zygotes form. Amenorrhea is basically, when it ceases, okay? Menstruation or menses, that's when your progesterone levels and estrogen levels start to drop. When in the uterine cycle, progesterone levels at their highest. Yeah, secretory phase. Secretory phase. Which of the following statements is true of both the male and the female reproductive systems? Which is true for both of them? Remember, the urethra is part of the, yeah, C is correct. That's right. Remember, gametes start before birth in females, okay? And they start at puberty in males. The urethra is part of the reproductive system. That's only true for males, not females. And the primary sex organ is found in the abdominal cavity. That is true for females, but not for males, okay? So, but for both, they're formed by meiosis. The areola is an area found on the what? And the reason the areola is hyperpigmented, it's hyperpigmented so it's darkly pigmented so that the newborn, the neonate with very poor vision at that stage of his or her life can see the nipple and start suckling. And start suckling. For food, okay? That's what baby wants is food, okay? It's amazing how everything works together. I mean, nothing's by mistake. There's no mistakes in human. Crossing over occurs during which phase of meiosis? Yeah, prophase 1, that's right. Prophase 1 of meiosis 1. Which is the following has a very acidic pH? Yeah, this is extremely acidic. Okay, it is mainly because of the glycogen that's secreted, because glycogen and the bacteria love glycogen. Okay, and they produce an acidic pH. You know, those would be the useful bacteria, though. Okay, but it will help destroy the bad bacteria. Cryptorchidism is a condition in which the testes fail to descend. It's usually at that last trimester. Okay, one of the testes fails to descend, and that makes the male infertile. It causes infertility. Remember, you have to have 40 to 750 million. 40 to 750 million sperm to be fertile. Which the following structure in the female are homologous to the scrotum? That would be the labia majora. Let me try another one here. That's not the one I wanted.
Professor Morgan,
Yeah.
there was a notification sent out
Uh-huh.
final exam being on May
Uh-huh.
12th and due at 9 p.m. Is that a different test from ours or?
Yeah, it is.
Okay, because I
Yeah,
want.
that's the other class. That's the other teachers. That's the other teacher that does the other thing. I think Fitzgerald has hers done it by 9 p.m.
Okay, yeah, I thought so, but I just wanted to make sure.
Yeah, no, yes, that's not me. Yeah, I'm gonna try to change mine. Okay, let's look at the urinary system. Okay, here we go. The tube that carries you, okay, that's going to be the what? Ureter, The renal medulla is also called the, right. The space inside the kidney is the renal sinus. The renal pelvis or the sinus, actually the pelvis is the collecting structure, and they call that space the renal sinus, but uh, it occupies the same area. Okay, which of the following empties urine directly into the renal pelvis? Here you have to know that would be the major calyx. That's right. Remember, minor calyx drains the pyramid. Minor calyx is your first urine draining structure. Minor calyx is your first urine draining structure. Which drains the renal pyramid. Okay, and then the minor calyx empties into major calyx, which empties into renal pelvis, which empties into ureter. That leads to the bladder. That leads to the urethra. Those are your urine draining structures. Which of the following vessels will be found in the renal columns? Well, remember the arcuate artery is between the cortex and medulla. Yeah, C is correct. Nephrons are found mostly in the renal cortex. Remember, that's where you find most of your nephrons and your blood vessels and renal corpuscles. Renal corpuscle consists of two things. Yeah, D is correct. Which of the following structures are not part of the nephron? All these are part of the nephron except for the collecting duct. Collecting duct is not part of the nephron. Podocytes are part of the glomerular capsule. That's right. The visceral layer of the capsule is part of the filtration membrane, right? Filtration membrane. Which of the following conditions would increase the GFR? Increase in colloid osmotic pressure. That would actually decrease it. Okay, that would actually decrease. An increase in COP would actually decrease the GFR. Agree? Yeah, definitely an increase in glomerular. This would also decrease it. Okay. So B is correct. Okay. Which of the following substances would not be found in normal filtrate? Remember, you'll see glucose, sometimes potassium and urea, but you should not see albumin. Okay, so that's the answer. That's the answer. Albumin should not be seen. Which of the following would be a result of an increase in systemic blood pressure? Remember, this is what we call the myogenic effect. Yeah, that's when you get an afferent arterial constricting. Remember, if you remember it like this, when your blood pressure increases, you need to protect the capillaries inside the glomerulus. So why the blood vessels do that is they reduce blood flow in the glomerulus. Okay, the only way they can do that is to constrict the afferent arteriole. Remember, constriction of the afferent arteriole is like turning down the faucet to your sink. Remember, if you think of this, the glomerulus as let's say a sink in a kitchen or a bathroom. The glomerulus is the base of the sink, okay? And the faucet is the afferent arteriole, and then the drain plug that lets the water out of your sink is the efferent arteriole, okay? So when you turn on the faucet, you get more water in your sink and it fills up, and if you put the drain plug in, you keep the water in your sink, okay? So constriction of the efferent arteriole keeps more water in the sink or keeps more blood in the glomerulus. So you're basically turning down the faucet of your bathroom sink and reducing the water flow into the sink by constricting the afferent arteriole. It's the same thing as turning down your faucet that goes to your water in your sink. It releases the water from your into your basin, into your sink. That's the best way I've used to remember how the glomerulus works. Okay, if you think of it as a kitchen sink and the faucet being the afferent arteriole and the drain plug as being the efferent arteriole. Okay, and so it's very similar to what you do in a sink when you're trying to get rid of the water in the sink or increasing the water in the sink. If your GHP is 60, COP and CHP are 25 and 35, what would be the net filtration pressure? Well, for that one, you just have to remember your net filtration pressure, NFP, is equal to the GHP, your hydrostatic pressure, minus the other two forces added together. So your COP plus your CHP. CHP stands for the capsular hydrostatic pressure. Okay, so you take 60 millimeters of mercury minus 35 millimeters of mercury plus 25 millimeters of mercury, which equals 60 millimeters of mercury minus 35 plus 25 is 60, minus 60 millimeters of mercury. So 60 minus 60 equals 0 millimeters of mercury. Okay. The answer would be A. And that's how you figure out any net filtration pressure. You take your glomerular hydrostatic pressure, which is basically going in the forward direction, and you take your colloid osmotic pressure added to the capsular hydrostatic pressure, and that would be 35 plus 25. So 60 minus 60 is 0. Okay, so that would be a diseased kidney because it's got a net filtration pressure of 0. So that means the kidneys are failing. Okay, you're in renal failure. You know, I never have seen a CHP 25 millimeters of mercury. A normal CAP at the most is about 10 millimeters of mercury. So this is just hypothetical, and that would be only that you wouldn't have if your kidneys were failing. Your capsular hydrostatic pressure could be 25 because all the fluid is seeping out of your glomerular capillaries because they're not working anymore. The filtration membrane is not selective anymore. But anyway, you wouldn't have any filtration pressure at all if you had those values. More typical GHP is about 55. GHP is normally about 55, 50 to 55. And a normal colloid, they call it COP, but it's actually the glomerular colloid osmotic pressure. So the glomerular, they really should say GCOP. A normal GCOP is close to about 25 millimeters of mercury. Okay, that's a normal GCOP, and a normal capsular hydrogen. GCOP stands for glomerular capillary osmotic pressure. Glomerular capillary osmotic pressure, and it works opposite of GHP, opposite of GHP. And the capsular hydrostatic pressure, CAP, is usually at the most 10 millimeters mercury. Which of the following will reduce the GFR? Well, increased GHP would increase it. Okay, ADH and aldosterone really aldosterone would cause sodium water reabsorption, so that would in fact reduce it also. Okay, the only one that would increase the GFR that would reduce the GFR would be angiotensin 2. Okay, because you're constricting the afferent arteriole, and you're turning down the faucet to your sink, so you're reducing water to your sink, or you're reducing blood flow to your glomerulus. Okay, so you're reducing the GFR. So angiotensin 2 is the only one that would reduce the GFR because it causes vasoconstriction of the afferent arteriole. turning down the faucet. Turning down the faucet of the sink. Most of the nutrients in filter are reabsorbed where? Correct. Good. The distal tubule, a blanket secrete for every sodium. Potassium, yeah, potassium is right, plus hydrogen. Hydrogen is also secreted for every sodium. When glucose spills over the urine, it has it's basically exceeded its transport maximum. Remember, when you have diabetes, you basically, your glucose will not be transported into the cell because the insulin is either not being produced or the receptors on the cells are not sensitive to insulin. They're resistant. Okay, either way, your glucose will start spilling out into the urine because the transport maximum, all those transport proteins are saturated, okay, because you got too much glucose in the extracellular fluid. And therefore, too much glucose, you're going to saturate all your transport proteins, so the glucose will start spilling into the urine, okay? If your urine contains fewer potassium than your filtrate, then the potassium has been what? Well, that means you got it more in the plasma than in the filtrate. So that means, yeah, that means it had been reabsorbed. As it went through, it must have been reabsorbed back into the blood. Which of the following is actively transported out of the loop of Henle? Sodium. Remember, active transport out of the thick segment. Remember, it's actively transported out of the thick segment. The ascending limb. Remember, water is reabsorbed out of the descending nephron. That would be the thin segment. But that's passive diffusion. Which of the following would be an abnormal constituent of the urine? Well, you're going to see sodium. Yeah, C is correct. The collecting ducts are permeable to water when the hormone ADH is present. Okay, that's just one of the things you have to remember. Which of the following would be an abnormal pH? Remember, the pH will vary between pH. You usually have a range. pH range is 4.8 to about 8. Okay. So 11 would be out of the ordinary. Okay. 11 would be an abnormal pH. The urine is slightly acidic, though. Okay. The portion of the nephron that maintains the hypertonicity of the renal medulla is the hypertonicity means high osmolarity or osmotic gradient of, around, the maximum hypertonicity is 1200 milliosmoles. And this is maintained by the nephron loop. Because it is differentially permeable. Nephron loop is permeable in the descending limb, but not in the ascending limb, and permeable to salt, which is basically we're talking about sodium chloride, in the ascending limb, but not in the descending limb. That's what we mean by differentially permeable. You have these unique characteristics of the nephron loop in different parts. But what that does, it creates a concentration gradient. So the deeper you go into the medulla, so the deeper that you travel into the medulla, the more concentrated the interstitium concentrated the renal interstitia and the tubular fluid is. And that creates a concentration gradient, okay? So it's the nephron loop, okay? Because of its differential properties, those unique properties that make it permeable in the descending limb to water, but impermeable in the ascending limb. And permeable in the ascending limb to salt, but impermeable in the descending limb to salt, okay? And what that does, that builds up a concentration gradient deeper into the medulla. That's because you got those unique properties to the nephron loop. The main function of the nephron loop is to concentrate the urine. The main function the primary purpose primary function of the nephron loop, or loop of Henle, is to concentrate the urine. Okay. So when urine enters the collecting duct, it would be hypotonic. It would be actually hypotonic. Okay. It's usually hypotonic, which means it's usually about 50 to 100 milliosmoles, okay, by the time the tubular fluid gets to the collecting duct. Reaches the collecting duct, the osmolarity is 50 milliosmoles, 50 milliosmoles. you know, plasma is, about 280 to 300 milliosmoles. Okay, that's normal osmolarity, okay? So it's going to be hypotonic. What part of the nephron employs the countercurrent mechanism? Countercurrent means blood is traveling in one direction like in the vasa recta, and tubular fluid is traveling in the opposite or counter direction. Okay. So not only that mainly occurs in the juxtamedullary nephrons. Okay. It occurs in the juxtamedullary nephrons. In the nephron loop of Henle. That would be the loop of Henle. Okay, the loop of Henle in its association with the vasa recta. Remember, the vasa recta is that blood supply. So here's your nephron loop coming down here like this. Here it's coming down from the cortex, parts tubule, and then here's your nephron loop coming down like this. Makes a turn, then becomes the ascending limb. Then it becomes a distal tubule. Then it enters the collecting duct. Okay. And here's the collecting duct out here. Okay. So your tubular fluid is going in this direction. Going toward the collecting duct. Okay. Where your vasa recta is mainly traveling in the opposite direction. Here's your vasa recta, which is intimately associated with the nephron loop. Okay. So let's put, so here's your vasa recta, which are your capillaries that basically are parallel to the nephron loops. So they're going to run like this. So here's your vasa recta. Okay, so this blood flow is going in this direction. Okay, this will be the descending limb of the vasa recta. Then it makes a curve and comes out the ascending limb in here. Okay, so it's going in this direction. Okay, so you got your tubular fluid going in one direction and then the blood going in the opposite direction. Okay, so that's what we call countercurrent. Okay, and what that does, it allows what's being pumped out of the nephron loop to be absorbed in the vasa recta. Okay, and remember, water is being reabsorbed in the descending limb of the nephron loop on this side. Okay, this is your descending limb of your nephron loop. So water comes out on this side, but then it's going to be reabsorbed by the vasa recta. Okay, so the vasa recta is an intimate association with the nephron loop, and that basically serves to conserve water when you're dehydrated. Okay, it conserves the maximum amount of water. That's why you can produce an extremely concentrated urine when you're extremely dehydrated. Okay, because the vasa recta will absorb every bit of water that's reabsorbed by the nephron loop. Okay, because they're way down here in the kidney interstitium. That's the reason you have juxtamedullary nephrons. Even though they're not very many, they have these vasa rectas that can absorb all that water back from the nephron loop. The cells of the macula densa and the JG cells form the what? The JGA, the juxtamedullary apparatus. What maintains the potassium concentration in the cytosol? That's your sodium potassium pump. An elevated sodium concentration is what? What do we call that? Yeah, hypernatremia. Hypernatremia. Which of the following happens during compensation for alkalosis? Well, remember, alkalosis means a pH greater than 7.45. That's all alkalosis means, a pH greater than 7.45. Okay. So if that's happening, you need to get rid of your bicarb, because bicarb is a base. You want more hydrogens when your pH is high, okay, to bring it back down, because you don't have any hydrogen. Okay, you don't have as much hydrogen. So the kidneys will excrete bicarb. The normal pH is what? Correct. So what if the PCO2 in the plasma increases? What effect will this have on pH? Will the pH increase or decrease? Yeah, the pH will decrease. Remember, when PCO2 goes up, that means your hydrogen goes up. Remember, CO2 plus water generates carbonic acid, which then immediately dissociates in the hydrogen ion and bicarb ion. At your equilibrium. So when CO2 goes up, your hydrogen goes up because of that equilibrium. And then there's an enzyme that I must mention that occurs here also. There's an enzyme that occurs right here called carbonic anhydrase. There's an enzyme called carbonic anhydrase that facilitates that conversion inside your red blood cells. Okay, but once that carbonic acid is made, it dissociates into hydrogen and bicarb. Remember, this is carbonic acid. This is a weak acid. And this is your bicarbonate ion, which is a weak base. Your carbonate is basically your base. Carbonic acid, misspelled that. Carbonic acid. Carbonic acid. So the pH would decrease, okay? That's an inverse relationship. The pH would decrease. As the CO2 goes up, that's the reason when you actively exercise, you have active metabolism, you're producing more CO2, the pH goes down, that causes you to breathe faster, okay? Because your pH is dropping. A patient presents with severe metabolic alkalosis. Now, if it's metabolic, remember you're looking at the bicarb levels. You're referring to the bicarb levels, HCO3. Respiratory, you're looking at the carbon dioxide levels. Bicarbonate refers to metabolic source or the acid-base condition. So the appropriate respiratory compensation would be, so if you're in metabolic alkalosis, that means your pH, by definition, is greater than 7.45. That's what alkalosis means, okay? And it's not due to a respiratory condition, but it's a metabolic condition that's causing the alkalosis. So, but the respiratory response would basically try to eliminate the excess bicarb. And the only way you can do that is to slow the respiration rate down to accumulate more CO2. And when you accumulate more CO2, you accumulate more hydrogen. And that's what you need. You need more hydrogen to neutralize the excess base. So it would be a decreased respiratory rate, okay? That would be the answer, okay? A decreased respiratory rate. Just like in metabolic acidosis, you'd have an increase in respiratory rate. In metabolic acidosis, the respiratory compensation would be an increase in respiratory rate. Okay. But anyway, it's a decreased respiratory rate. Okay. What's the most abundant intracellular cation? B is correct. A nurse is asked to start an IV line of isotonic. This is too real here. Of isotonic 0.9% sodium chloride. He accidentally grabs a bag of hypertonic 0.9% sodium chloride solution instead. What would happen to the patient? 9% sodium chloride, remember, is very hypertonic. Okay, and basically what you're doing, you're simulating dehydration to the patient. You're dehydrating the patient basically. So what would happen to the patient's fluids? The patient's cells will shrivel due to losing water by osmosis from the cytosol into the hypertonic extracellular fluid. Okay, so that's what would happen if you gave him 9% sodium chloride solution. Okay, that's called crenation. Okay, the cells will shrivel. The red blood cells. The red blood cells will look like this. Okay, instead of round, they'll look like this. They'll be crenated. Okay, they should be round like this. It should be elliptical, like that. Okay, but they'll look like this. That's called crenation because they're losing water. Because cells are losing water to the hypertonic fluid. Surrounding it. The vasa recta is labeled what? Correct. That's correct. The arcade artery is labeled what? Correct. Mm-hmm. The periturbocapillary is labeled what? That's right. The efferent arterial is labeled what? Yeah, it's labeled C. Yeah. See the efferent arterial, you can see it's coming out of the glomerulus. Here's the glomerulus right here. And so here's the afferent and here's the efferent. Okay. And then it becomes the vasa recta. This is a juxtamedullary nephron. So the efferent arterial is becoming the vasa recta in your juxtamedullary nephrons. Okay, but in your peritubular, in your cortical nephrons, the efferent arterial becomes the peritubular capillaries. Okay, but in your medullary nephrons, it becomes the vasa recta. Okay, and then they drain into the interlocular vein. Okay. What's the afferent arterial label? So the hormone identified by the question mark up here, coming from the anterior pituitary and going to the testes, which of these would be the hormones that would be present here? That's right. B is correct. All right. Well, I guess that will be it for today. And I'm going to try to change the date for the final. All right. And it'll be the same day, but I'll try to give you a longer window period.
I have a question.
Okay. Sure.
Do you give extra credit for filling out the evaluations?
For the evaluations, sure, especially if it's a good one. I will give extra credit. Yeah, if it's like, I also look at attendance, that kind of stuff. If it's real good attendance, I usually, like if you're like a point away or so, and you're short like 1.5 points or something like that, I'm going up to grade.
Like we could send you the screenshot
Yeah.
of the evaluation that we took, and then
Okay. Yeah. All right, guys. Well, I'll let y'all enjoy studying, I guess, and good luck. Yeah, that would be fine. Okay. Y'all have a great day and a good week, and enjoyed having you. Y'all are a good class. Mm-hmm.